Great. So finally, final talk this afternoon is from Matt Hoare, who's going to tell us why patients with cirrhosis get cancer. So I'm sure you all know Matt, university lecturer in hepatology here in Cambridge, who also leads, together with Victoria Snowden, the HCC service, and he's done considerable and fascinating research on liver cancer. So I think we're very lucky to hear him this afternoon. Matt, over to you.
Okay, thank you. And George, can you see me and hear me okay?
Yeah.
Yeah, lovely. Okay, so as part of my work on HCC, one of the questions that really sort of motivates me in what I'm doing in the research lab at the moment is to try and consider why patients with cirrhosis get cancer. And you might think this will be a fairly obvious thing and that would have been worked out by now, but really we don't have a good handle on why patients get HCC in liver disease as they do. Most of you will be aware that in general things are going in the wrong direction with liver cancer. So this is data from the US, and all the predictions are that there is a rising incidence of HCC in the US population, and all the money is going on. If you believe pollsters, that primary liver cancer will become the third biggest cause of cancer-related death in the US by around 2030, just at the same time that many other cancers are going in the other direction. And you can see this in this rather fascinating slide, which you can dig out of this website here. What they are charting is the 5-year survival rate for all types of different cancers from the 1970s here in the pink dots and currently here in the blue dots. And what you can see is there's been enormous progress made in a whole range of different solid organ malignancies, such as prostate and breast and melanoma, enormous progress made in the hematological malignancies, such as lymphoma and myeloma, where you get these big increases in 5-year survival. But yet liver cancer is down in this sort of inglorious bunch of aerodigestive cancers, such as pancreas, esophagus, and lung, whereby we've made precious little progress over the last 40 years, and the outcomes remain fairly dismal. But it shouldn't really be like this, because unlike most of these other malignancies, we in hepatology have a well-recognized pre-malignant state. We have an at-risk population. We see them in our clinics every single week. So we know that if you have a normal liver, then your risk of getting an HCC is pretty low. We know that if you then abuse your liver, that you transition from this state to this state, and that your risk of HCC goes up dramatically. And so that if you are a middle-aged smoking male who is obese, then your risk of developing HCC approaches 4% a year. But not only this, that we also recognize the phenomenon that you can see livers with multiple synchronous primaries, such as this one. I mean, this is one of our explants that we took out, and obviously we knew about this HCC, which had undergone embolization. We knew about this HCC, but what we didn't know were about the 4 other HCCs, one here, one here, one here, and one here. And I don't need to show you complex genetic sequencing or other scientific techniques. You can see that all these tumors here are all different colors. You know, I don't even need to show you microscopy. You can see that each one of these cancers is going to be a completely distinct molecular subtype. And not only do we see synchronous primaries in patients with liver disease, we also see metachronous primaries. And so whereas we would see this as clinical hepatologists, as an at-risk population, as a cancer biologist, we would see this as a cancerization field effect, such that the entirety of the liver has been affected by something which increases your risk of getting a tumor from any part of it. And so early detection is all very well in other types of malignancy. You know, we might have been able to detect this one earlier, but if we're looking for something that's specific to this cancer, you're going to miss all of these other different cancers within the same patient. And so early detection isn't really going to work in patients who have liver disease. We need to be moving this a step forward again to think about early prevention. But if we're going to think about early prevention of HCC, we need to understand why patients with cirrhosis get it. Now, if we look at the genetic variants, which we know operate within liver disease and chronic liver disease in particular, we know that if you start out with SNPs, single nucleotide polymorphisms of certain loci, particularly PNPLA3 amongst several others, or you have variants in coding genes such as the HFE gene, you increase your risk of developing liver disease, of progressing with liver disease, and then getting a tumor on the background of liver disease. We know what happens at the other end. If you have one of these cancers, as I'll show you in a minute, with the very big, large-scale sequencing studies, which we know that the kind of mutations that you're going to see from these tumors are mutations in the TERT promoter, mutations or losses of p53, or alterations within the beta-catenin pathway. But really what the big gap in our knowledge is what happens between these 2 points. What is it that's going on which encodes this cancerization field effect? What is it that underpins this at-risk population? And so if we could really understand that, then we could either try early detection, or better, we could go for early prevention. So about 5 years ago, we teamed up with the guys at the Sanger, and they had developed a relatively new line in sequencing normal tissue. Now, you'll be well aware that we've been sequencing tumors for many, many years now. And doing genetic sequencing of tumors, whether you're going to go for targeted sequencing, whole exome sequencing, or whole genome sequencing, is relatively straightforward because you know that a cancer is at some level inherently clonal. Sequencing of normal tissue is rather more difficult. You'll be aware of taking a buccal swab or a buffy coat to get someone's genotype. But what you're getting there is you're getting a summation of their average, what their germline code looks like. If you want to look at what damage has been accrued during life, that's actually very difficult because normal tissue is such a patchwork of small little clones of cells which can accumulate damage. And if you bulk sequence them, you're just going to lose it in the data. So they started out by looking at sunlit exposed eyelid skin. And this is a good place to start because obviously elderly patients have eyelids tightened up as part of an ectropion removal. And so what you can get is that you can get skin from relatively elderly patients. And then what you can do is in this study, this is many years ago now, that they did physical microdissection. And they took these tiny little punch biopsies and then did whole genome, did targeted sequencing of cancer genes. And that this is a 1 millimeter square piece of skin, and that each one of these circles here represents an individual clone of cells inferred from the sequencing data. And if they are colored, they contain a genome variant or a mutation which has previously been associated with cancer. So this is phenotypically normal eyelid skin. There's no cancer here on histology. And yet when you sequence it, you find that there are losses of p53, there are gain of functions of Notch and FGFR. So this was perhaps a surprise at the time, that phenotypically normal tissue could contain the same genomic changes that you see within the cognate cancer. And that this has then been replicated in a series of other tissues. This is esophagus, and these were tissues that were provided by Karosh to the Sanger team. And what you can see here is that this is young, this is old, and that you're going through life as we go down the page, and that they've applied the same methodology here. And you'll see that if you are young, you have small clones, but you still contain potential oncogenic drivers even at the age of 20. And that by the time you're 70, you've got, in phenotypically normal esophageal mucosa, you have an absolute bucket load of mutations which would normally be associated with cancer within that tissue. So we hypothesized that you would see something similar within diseased livers. And so what we did was that we took patients who were either undergoing hepatic resection for colorectal metastases, and they didn't have any other liver disease, and that we were sequencing the background, not the colorectal met. And that then we also took liver from patients who were undergoing transplantation for alcohol-related liver disease or NAFLD, and that these were really an end-stage phenotype, because we thought what we needed was patients with an extreme clinical phenotype. So they were patients who had an HCC as well as liver failure. And what we did was that we took, here's the HCC here, and that we took cause well away from the HCC in order to sequence the background liver. And then what we did was that we then got frozen sections, we performed laser capture microdissection, and then whole genome sequencing in order to try and figure out what was going on within in this chronically diseased liver tissue. And what we found was really quite fascinating. So if we start with the normal liver here, so this is a 65-year-old male undergoing an hepatic resection for colorectal metastases. Each one of these little crosshairs here is a laser capture microdissection. And then over here, what we have is each one of these maps onto one of these little stalks here of this phylogenetic tree. And then what we are plotting here is the number of single nucleotide variants. And what you can see is that all of these microdissections come from individual clones. There is no relatedness between these clones, i.e. they all continue in one solid little line, except for this one. And what you can see here is that clone 18, which is just over here, it's a bit difficult to see, then begat two separate clones of clone 3 and clone 25. So clone 3 and 25 shared all of these mutations, but then had these individual mutations. So the inference from this is that normal liver is inherently polyclonal, lots and lots of unrelated little clones which have not shared a common ancestor, probably since the outgrowth of the foregut. If you then look at cirrhotic liver, the situation is completely different. So what you can see is probably best seen here on the oil red O staining. You've got these swathes of fibrosis and then these cirrhotic nodules. And that then when you start performing individual microdissections, what we then found was that most cirrhotic nodules were either oligoclonal or indeed monoclonal. So you see here, this large one just here, this large nodule here, only had clones which shared mutations with clone 28, i.e. that clone 28 arose first, and then you then had an outgrowth of a whole load of other different clones, i.e. that this nodule here was monoclonal. And similarly, there was a nodule up here, which was mostly monoclonal, and likewise with most of the other patients. So what you are seeing is that these cirrhotic nodules are oligoclonal or perhaps monoclonal in many cases. The mutations are not shared across fibrotic boundaries, and so each cirrhotic nodule is its own genomic microenvironment. And that then within a cirrhotic nodule, you then get subclonal diversification, i.e. you get proliferation, and then you get the acquisition of further mutations. I explained before that many large-scale data sets, and this is just the TCGA data set from a while ago, have profiled what the driver mutations are in HCC. Now, this is an oncoprint. Each column is a patient, and then each row is a genomic loci which is mutated. And so what we know from hundreds of patients is that around a third of patients with HCC, their HCC will see either a mutation or loss of p53. Around a third will see either a mutation or activation of beta-catenin, and then there is a long tail of other mutations. And so what we were expecting to find when we started looking for locus-specific damage within chronic liver disease was to see some of these mutations. And so this is a similar oncoprint. So these are the 30 loci which are most mutated from the TCGA dataset. And each one of these columns is an individual microdissection. So these are the 5 normal patients. And what you can see is actually the first surprise was that in 60 to 70-year-old, mostly male patients undergoing resection for colorectal metastases is that we saw very, very few cancer-associated mutations. What was a bigger surprise was that then if you took patients who had end-stage liver disease and HCC and were undergoing transplantation, is that they also did not have any cancer-associated mutations within their background liver. So this is a complete surprise and means that liver is completely different to skin, esophagus, endometrium, and blood, most of the other tissues of the body. And so you can make concrete assertions from this. So we know that less than 3% of samples had a driver variant, that the cancerization field effect is not encoded at oncogenic mutations which are seen in HCC, but more importantly, more sort of globally, that actually there is no positive selection pressure to acquire these cancer-associated mutations in the context of chronic liver disease. That's a pretty profound finding. So that if you were to lose P53, that provides no additional selection pressure, that the regeneration mechanisms within the human liver are so perfect that mutations which would cause activation of beta-catenin or MYC or any of these others to provide no additional selection pressure. So this is quite a cool finding, but it doesn't really take us any further forward in our original question as to what on earth is going on. How is this cancerization field effect encoded in chronic liver disease? And so what we've done now is that we have gone further. So the original data set that we published was around 400 odd whole genomes, that we've now expanded this. We've now done about 1,200 whole genomes. There's actually now, from the TCGA, there's only one bigger data set than this now, and that's breast cancer. And that we've now done this in chronic liver disease. And that this now encompasses 5 normals and 27 patients who are either going resection or transplantation for NAFLD or alcohol-related liver disease. And these are your patients, okay? So we have patients in here from Norwich, we have patients in here from Luton, we have patients in here from Southampton. These are your patients who are undergoing operation here. And so this is the disease which I'm seeing in my clinic and you are seeing in yours. And what we're finding now, that we now have the statistical power to look genome-wide at different loci, is that we're now finding recurrent mutations at only 4 loci, and that is FOXO1, GPAM, ACVR2A, and CLCN5. Now, none of that will mean anything to you, but what I want to do today is to talk to you simply about mutations at FOXO1. Now, the story is incomplete. We don't have all the conclusions, but the potential impact of what we have found is really, really profound, and we thought it was worth sharing it with you today. So what is FOXO1? Now, I realize at a clinical meeting it's dangerous to start talking about transcription factors, but FOXO1 is the dominant transcription factor which is immediately downstream of insulin. So in a low nutrient state where you have low nutrients and low insulin signaling, the insulin receptor is off. AKT at cytoplasmic enzyme is off, and therefore FOXO1 resides within the nucleus, driving gene transcription. If you then eat and your insulin level goes up, the insulin receptor then activates, it then transduces the signal through AKT, which then phosphorylates and removes FOXO1 from the nucleus and then prevents it executing its normal program. So what we hypothesized was that mutations in FOXO1 would prevent its removal from the nucleus. Okay, so let's see what we find. So within our cohort, so this is 27 patients with chronic liver disease, we have found mutations within FOXO1 within 1, 2, 3, 4, 5, 6 patients who have non-alcoholic fatty liver disease or alcohol-related liver disease. But we are not just finding mutations within FOXO1, we're finding mutations within a single amino acid of FOXO1. So this is the gene runs this way, whereas the amino acid code runs this way. And all of the mutations were at S, and that they were an S22W substitution. There was one patient with NAFLD who had an R21L, but we haven't found that in a separate patient. So we've got evidence of recurrent mutation within a single or 2 adjacent bases within a single transcription factor within multiple patients undergoing operation for chronic liver disease. Now, the next thing that we found was that within particular patients, we were finding this mutation multiple times. So you see that these are two patients here. They're both got non-alcoholic fatty liver disease, and as you can see from the scale bar, we're talking about a sort of 5 millimeter little chunk of background liver, and that this patient has 1, 2, 3, 4, 5 mutations within a 5 millimeter chunk. This patient here has 1, 2, 3 mutations. Now this is incredibly strong evidence for a phenomenon called convergent evolution. Now, convergent evolution suggests overwhelming positive selection pressure to acquire something. And it's best explained through normal organismal evolution. So if you think about creatures which have evolved over time, that we can see convergent evolution through the acquisition of wings. So you can see that this has been evolved in birds, this has been evolved in mammals and bats, and also within insects. So the selection pressure to acquire the ability to fly through a wing is overwhelming, and therefore positive selection pressure has driven the acquisition of this trait on multiple occasions. What we are seeing within the context of chronic liver disease is evidence of convergent evolution within the same liver. So what you can see from the phylogenetic trees are you've got multiple independent clones of hepatocytes within these livers which have acquired the same mutation. And therefore, this positive selection pressure to acquire this mutation must be overwhelming within these patients. What we've then done is we've gone on to demonstrate in vitro that if you model these mutations, that these mutations are actually functional and do what we expect them to do. So here, this is the HepG2, this is a liver cancer cell line, and what we have done is that we have taken wild type FOXO1 with a fluorescent tag to GFP here, and if you believe what I just told you on that cartoon a second ago, what we're expecting is that when you starve the cells, it should be nuclear like this, and then you add insulin, and it then promptly leaves the nucleus like this. Whereas if you put the same construct into liver cancer cell lines with the mutation, what we find is that it's in the nucleus, and then you stimulate them with insulin, and it remains within the nucleus. And you can see that here in this live cell imaging. So we know, at least in vitro, that these mutations are functional and drive a phenotype of insulin resistance. We've then gone back to serial sections from our patients, and we've then done further laser capture microdissection, pulling out little clones here in green, which are wild type, and in blue, which are mutant for the FOXO1 mutation. And this is very, very recent data. And what we have found using RNA sequencing to look at what these cells are doing is that we're finding that there's big, big changes in their mitochondrial gene pattern, and particularly within oxidative phosphorylation within these clones. So it seems to be that there is a overwhelming pressure to acquire mutations within FOXO1, within chronic liver disease, and that it happens on multiple occasions in a sub-cohort of patients. But why? At this time, we don't really know, because it must have a positive gain for these cells or for these clones, but this is clearly going to require an awful lot more work. But clearly for you and me, from a clinical point of view, we have to ask, well, does it have diagnostic or prognostic importance? If we found this within patients, is it a good prognostic or a poor prognostic sign? At the moment, we just don't know. And whether this is tractable, again, we don't know. But I think you can agree that it's an incredibly exciting finding, and one in which we can hopefully explore over the next few years. So in summary, HCC has a clear at-risk population. We all know that. Early prevention has to be the way to go rather than early detection. We have found convergent evolution of mutations within FOXO1 in chronic liver disease, which are functional. Hopefully, over the next few years, we'll be able to figure out what that means and what we can do about it in patients. So it just leaves me to thank particularly the guys from the Sanger who've done most of the sequencing work, but particularly all of the people who have funded me over the years. Thank you very much.
Well, thank you, Matt. That's a really wonderful piece of work, and it's nice to see where it's up to. There will no doubt be some questions coming through, but I wonder if I can just kick off just by maybe a slightly challenging one, which is your statement that it's better to prevent cancer. And where do you see this latest work, this potentially driver mutation in FOXO1, as helping you to identify patients who need somehow more intensive prevention strategies? And indeed, where do you see the previous work about the somatic mutation burden or somatic mutation signature as identifying patients who need more intensive prevention strategies versus, by the way, preventative strategies for any patient at risk of developing cirrhosis in the future?
Yeah, well, I mean, firstly, what we need to do is to try and go earlier in our sequencing strategy and try and work out whether this is a linear acquisition through people's progression to chronic liver disease, or does it occur in a non-linear fashion? Is it people who are acquiring mutations quicker that are going to do worse? We don't know, because obviously at the moment we have taken patients with the extreme clinical phenotype on the supposition that we would have the sort of the highest gain to find. In terms of how we could potentially translate this, in terms of the older work, one of the things that we want to try and develop is to look for circulating DNA. Now, you'll be aware of circulating tumor DNA when people with established cancer. Well, we know that you can detect circulating cell-free DNA, so non-malignant derived DNA from the liver, and that there are strategies to try and identify, based on methylome sequencing, whether it came from the liver. So if we could potentially do shotgun sequencing of cell-free DNA within people's peripheral blood, look for liver derivation, and then look for mutational burden within it, that could be one strategy to avoid the need for biopsy. In terms of the later work with FOXO1, I mean, I think that really opens up some potential avenues, because obviously if we can try and figure out what the metabolic correlate is of this gain of function, because if we've got 6 mutations within a 5 millimeter chunk of liver, therefore if we were to bother sequencing the entire liver, you would suppose that there would be hundreds to thousands of clones with a similar mutation within it, and therefore you would have a detectable metabolic effect peripherally. There might be some blood lipid or some carbohydrate that is a byproduct of a deranged metabolism that one could detect in the plasma. And that's certainly one thing that we could potentially do. I mean, at the moment, one of the main barriers is that we just don't know what the prognostic import is. Why is there such an overwhelming selection pressure to acquire this mutation? And why is it not found in the same cancer? So we're finding it in the chronic liver disease where it's never found in the same cancer. So it must have some benefit to the liver. Does it keep the wheels on the wagon for longer? Is that why the liver acquires this mutation? At the moment, we just don't know. But we can say that it is important based on the fact that it is repeatedly seen across patients, repeatedly seen within the same liver.
Yeah, so it has potential to be a very important biomarker. And there's a question that's just come through from Tess Cacciatolo. Is there any evidence that people developing HCC might become more insulin resistant?
Not to my knowledge. I mean, that would be, I mean, that would require obviously pre-cancer development stringent metabolic testing to see whether they were less insulin resistant, and then demonstrate that as they develop a tumor, then more. I mean, if we could develop this East Anglian liver registry and do, detailed metabolic phenotyping at enrollment, and then phenotype them as they develop tumors, that would have to be the way to go. We would have to do this prospectively.
Yeah, and as an extension to her question, actually, the next part of her question is, could insulin resistance predict FOXO1 mutation? And I have a question related to that, actually, is have you managed to identify any clinical characteristics which predict FOXO1 mutations?
Yeah, I mean, these are great questions, and obviously ones which we've already asked ourselves. The problem is that the technology to do sequencing of normal is fiendishly expensive. I mean, this is quite a large wodge of cash to do this study, and we have still only managed to do it in 5 normals and 27 liver disease patients. And then we found the mutation in 6 actual liver disease patients. So obviously, if we're trying to look then versus HbA1c versus central obesity or total obesity or triglycerides, or we're really p hacking here, and that what we need to do is to try and come up with an easier way of finding this mutation. And that we are trying to come up with some new methods of doing that, so that we can really expand this out to ask, across hundreds of patients, do they have a FOXO1 mutation? And then we can then adequately power the multivariate studies, which are going to need to be done to say FOXO1 mutation, triglycerides, or FOXO1 mutation, insulin resistance. At the moment, these are great questions and clearly need to be done. We don't know.
Well, Matt, it's really fascinating work, and I think we all look forward to hearing more about it in future, but I'm afraid time is pressing, so I'm going to hand over now to Bill to finish up the day. But thank you very much for that really fantastic talk.
Thank you, Matt. I'd just like to thank everyone, really all the speakers, co-chairs, all the participants. It's been a great attendance today, and in the nursing forum this morning as well. Thank you for all your questions and interest. I think it's been a great day. I'm glad the IT worked. I was a bit worried about that, but Will made it work. So thanks everybody, and I don't know what form this meeting will be in next year, but we hope to see you all. And just a final thank you to Rosie and Sue Bolton, who are our very capable admin team who organised all the registration and got you all here today. So have a good evening. Thank you again. Bye-bye.